AP Bio 4.1 is on cell communication. If you're an organism that's made of more than one cell, or you live amongst other cells, then you or your cells must be able to communicate and respond to signals. Cells communicate largely through chemicals, and when something is wrong with the transmission, reception, or response to the signals, you get issues. This is what the first part of Unit 4 is all about. So, how do cells talk to one another? One way is by direct contact, where one cell's surface proteins, called antigens, interact with another's. This is what your immune system mainly uses. The main way cells communicate is by releasing chemicals into their environment, which are picked up by other cells. Paracrine signaling is when the receiving cells are close by, endocrine signaling is when the receiving cells are far away, and chemical messages called hormones are sent through the blood or the lymph. Autocrine signaling is when a cell picks up its own chemical message. But right now, you are processing this information through synaptic signaling, where a neuron transfers electrical impulses from one to another or to a target cell through neurotransmitter molecules like serotonin or dopamine. 4.2 is the introduction to signal transduction. Cell communication can be broken down into three main stages. The first is reception, where a messenger molecule called a ligand binds to a protein in a cell's plasma membrane called a receptor. They are made specifically for one another like a lock and a key. When these two bind, it causes a change in the shape of the receptor, which initiates the second stage, transduction. Transduction can get complicated, but it basically takes the message from the outside of the cell and brings it inside so that the cell can produce the proper response to the signal. This works like a relay race where proteins called kinases phosphorylate or turn on others, which causes the phosphorylation of others and so on until the right protein or enzyme is activated to produce the response. This is called a phosphorylation cascade. Phosphatases do the opposite of kinases or turn off certain proteins. When a lot of responses are needed at the same time, second messengers are used, which are mass-produced molecules that can amplify a signal and activate a lot of proteins at the same time. The third stage is the response itself, which could make the cell grow, secrete certain molecules, produce proteins through gene expression, or just die. 4.3 is on signal transduction, where we'll get into a few more specifics about transduction pathways. A few types of receptors that'll be helpful to know are ligand-gated ion channels, which do exactly what their name suggests, and intracellular receptors, which are those located inside a cell and responsible for initiating a gene expression response. An important pathway to be familiar with for AP Bio uses a G protein coupled receptor, or GPCR. Epinephrine is a well-known hormone that produces the fight or flight response. When your body releases epinephrine, it prepares you for something potentially perceived to be life-threatening. Epinephrine receptors are GPCRs, so when epinephrine binds, it causes the activation of what's called a G protein, which in turn activates an enzyme called adenylyl cyclase. This enzyme produces a ton of cyclic AMP, a second messenger. CAMP then phosphorylates a lot of different proteins, initiating phosphorylation cascades that will ultimately produce the fight or flight response. One of these results is the hydrolysis of glycogen into glucose through glycogen phosphorylase. There's a lot more examples of this kind of stuff, so please watch the full 4.3 video on my channel. Topic 4.4 is on changes in signal transduction pathways. Our last two topics were about how a cell receives a signal, then sends it to the right part of the cell to produce the appropriate response to the message. There are a lot of moving parts in this process, and therefore a lot of places where this could go wrong. Mutations, which will come up a lot in this class, can alter the shape and thereby the function of a receptor, enzyme, kinase, phosphatase, or any other protein. And that can result in a cell over-responding to one signal and not responding at all to another. For example, P53 is what's known as a tumor suppressor protein, and when it's mutated, it cannot stop a cell's response to a signal telling it to divide, which leads to cancer. Or if insulin receptors are mutated, a cell can't store glucose from the bloodstream, and that leads to diabetes. The other thing that can mess up signal transduction pathways are certain chemicals like medicines or poisons. These could block a receptor, inhibit a kinase or enzyme, among a whole bunch of other things. Some poisons are really good at shutting down key enzymes like adenylyl cyclase or ion channels or pumps, and that can have huge consequences on cell functions, for better or for worse. Topic 4.5 is on feedback. You've probably heard this word before in school. If you get positive feedback on an assignment, you're doing things right. And if you get negative feedback on an assignment, there's something that you're doing that you have to fix. Your cells give feedback to one another on whether or not a change in your internal environment should continue or if it should be stopped. Keeping this internal balance is called maintaining homeostasis, and all living things do this through feedback loops. Here's an example. If you get cold, your body detects a deviation from your normal body temperature, and you shiver, among other things, to bring your temperature back up. If you're too hot, you'll sweat to keep your temperature down. These are called negative feedback loops. In all types of feedback loops, a sensor detects a change in the internal environment, a control center establishes a set point where a certain factor should be at, for example, blood sugar or temperature, and an effector gets told what to do by the control center in response to the initial change in the internal environment. A positive feedback loop will accelerate a physiological change in order to get something done fast. Cells respond to a change in such a way that causes more of the change, which creates a cycle. And that's kind of how babies are born. 4.6 is on the cell cycle. In order for multicellular organisms to grow or repair tissues or continue existing, cells need to divide, and this is their reproductive cycle. Eukaryotic cells spend most of their lives in interphase, which can be broken up into 3 parts. Gap 1 is where a cell carries out its normal functions and prepares for DNA replication. S is where it copies its DNA, and gap 2 is basically where a cell gets ready to divide into 2 copies of itself. M stands for mitosis, the division of the nucleus. DNA has been copied, and it needs to be split evenly between 2 daughter cells so that they are identical. Prophase, the first phase of mitosis, is when the DNA gets condensed and packaged into chromosomes. The centrosomes form the mitotic spindle, and the chromosomes get moved towards the middle of the cell to begin metaphase. Once all the chromosomes are properly aligned and the spindle is attached to the centromeres of all the chromosomes, anaphase begins and splits apart the chromosomes, which elongates the cell. This is followed by telophase, where the new nuclei are forming and the spindle gets broken down. Cytokinesis happens during and after telophase when all the other contents of the cell are divided. The cycle then begins again. 4.7 is on the regulation of the cell cycle. Cell division is a good thing, generally speaking, but too much of a good thing is a bad thing. The rate at which cells divide needs to be controlled. Not enough division means no growth or tissue repair, but too much division is also very bad. A cell's main way of controlling when it progresses through the cell cycle is through checkpoints. Basically, as a cell is preparing to divide, it needs to meet certain requirements before progressing to the next phase of the cell cycle. The proteins involved in these transduction pathways are called cyclins and cyclin-dependent kinases. For example, one type of cyclin activates the enzymes responsible for DNA replication. If something happens to one of the genes for a protein that helps continue or stop the progression of the cell cycle, then cell division can become uncontrolled. When this happens, a cell will typically receive a signal to self-destruct, which is called apoptosis. But when a cell can't properly respond to the apoptosis signal, it results in cells dividing rapidly and uncontrollably, which is cancer. A clump of cells that can't control how much they divide is called a tumor. We call it benign if it stays in one place, and we call it malignant if it finds a way to metastasize or move to another place in the body.